So about a year ago, my friend and neighbor Betty Friedman called and she said, you know, throughout this pandemic, so many people have been trapped inside. And she said, I've been thinking we're so lucky to have this beautiful river running right through our backyards. We should do something to help. Now, Betty's always right, as Ed will tell you. But she was really right about this, okay? We happen to live on Terry's Plain, and the river is our backyard, and it's wonderful. And that's something that we share. And so with Betty's call and some acknowledgement, we were off. So Betty reached out to the Farmington River Watershed Association. And FRWA's president, Michael Feldman, Michael, you out there? There he is. And Betty and Michael talked about what some interested citizens could do to help out with the FRWA. And that was the beginning. And so the Simsbury River people, our group of 8 neighbors who live here on the river in town, plus one very dedicated member of the FRWA board, and that's the Simsbury River people. So I'd like to introduce them very quickly. So it's me and my wife Christy, who's in the back, and our neighbors Betty Friedman and Ed Goldberg, and their neighbors downstream, Nick, who's in the back, Nick Gallick, and Emily. Emily's in the back with Christy. Our upstream neighbors, Susie Eblins and Donna Scala. Susie's in the back, and Donna. And our friend and great supporter from the FRWA board, Susie Barney. Susie's over here. That's the Simsbury River people as it stands. And so we all have this passion for the Farmington River. That's what we've got in common. We believe it's a treasure, and we're looking for ways to connect with fellow town residents in conservation through community. We're reaching out to our fellow neighbors to talk about how important this resource is to protect our river and to preserve it for generations to come. That's what we're looking to do. It's a treasure. So we had ideas about what would make a good Simsbury-specific river-themed program, and we reached out to Sally Rieger and the Lower Farmington River and Salmon Brook Wild and Scenic Committee. Say that 3 times fast, you win a prize. But we reached out to them to get their thoughts. And Sally said, you know, we've been looking for a forum for Dennis Quinn to come and talk about the research that he did on herpetology in Simsbury along the Farmington last summer. Well, when I heard that, I knew we had a match. That was perfect. So we said, we'll take the logistics and let's get Dennis to come out and talk to everybody. And today we're going to hear a little bit about the Wild and Scenic Committee's work. We'll hear from Dennis about his research, and David Sienisch about the beautiful Farmington River quilt that's around us. It was a great opportunity to be in this room with us out here. And we hope that afterwards you'll join us. There's still some cookies and Chex Mix, and there'll be some coffee left. And get to know some of your neighbors who share a passion about the river. We know you're here because you're interested, you're here because you care, and you're probably here because you want to make a difference. So welcome, and thank you for being here. And with that, I'll turn it over to Mike Ryan, who's the chair of, let me say it again, the Lower Farmington River and Salmon Brook Wild and Scenic Committee. Mike, thank you.
Thank you, Rick. You want me to...
Yeah. Will do. There you go.
All right. All right, thank you, Rick. Let me just get myself set up here. So again, I'm Mike Ryan, the newly appointed chairperson of the Lower Farmington River and Salmon Brook Wild and Scenic Committee. And I just can't thank Rick enough for setting this up, because I love the choreography of this meeting and the theme of the river, which usually is kind of one-dimensional, right? We're talking about perhaps our committee, perhaps another aspect of the river, but the way Rick really set this up with the river people, that was kind of new to me, but to understand that we have a group of impassioned neighbors living along the river, it ties in well to our committee, which is a committee of people that are really impassioned about the preservation of the scenic river we have here. And then to have Dennis here to give us kind of a slice of some of the real knowledge around the biodiversity that exists within the river, I think just really makes for a great presentation. So thank you for setting this up. So I'm just going to take about 10 minutes here. Some of you may be very much aware of the Lower Farmington River, Salmon Brook, and Wild Scenic Committee. For the rest of the program, I will just say Lower River Committee. But it really has kind of quite a history. And you know, I was new to it 2 years ago, and I can't believe really that it exists, how hard it was to kind of bring it into existence, and I feel really kind of honored to be a member of this committee. So where does it all really start? We kind of always start the presentation or the talk track off with the National Wild and Scenic Rivers Act that was passed in 1968 to protect the nation's greatest resources, right? Its scenic rivers and its wild rivers, which are really actually very distinct. There's wild rivers and there's scenic rivers. We actually happen to be in partnership as a scenic river, and they both have their specific designations. But between the two, there's about 266 rivers with that designation in the U.S. It covers about 11,000 miles of river, and there's 10 such designated rivers in New England. And it's really, as we'll talk about as we go through this presentation, it's all about once this designation is obtained, a committee is formed, and it really has two kind of primary missions: the preservation of the river and the education, or bringing people in as community members to help preserve the river and enjoy the river. So with regard to our river designation, it's kind of interesting. Many of you may know that the upper Farmington River achieved the designation of wild and scenic back in 1994. And that is not an easy designation to achieve. It's interesting, I finally met Dave, who is a member, one of the leaders of the Upper River Committee. He had started with that committee back in 1982, and they got their designation in 1994. Some of you, we have Sally Rieger in the room. She really, years ago, became impassioned around getting that designation for the lower Farmington River. And I'm not sure, Sally would have to tell you what year that started, but it was no small achievement, and it took until 2019 to get that designation. So you can see we've only had that designation for a little less than 4 years, and we've now set up an advisory committee, and that advisory committee is made up of members of the 9 towns. So if you look at this map here, you can see the purple line is the upper Farmington designation. The lower Farmington designation, which again was established in 2019, includes 9 lower river towns. As you can see, it's Burlington, Farmington, Avon, Canton, Simsbury. People sometimes are surprised when we say it includes Heartland, Granby, and East Granby, but that's because this designation also includes the Salmon Brook. So we have, and then obviously, as it goes out to the Connecticut River, it includes Windsor and Bloomfield. So there's 9 member towns. There's actually 3 member watershed associations. There's the Farmington River Watershed Association, the Quapabic River Watershed Association, and what is our third? The Salmon Brook Watershed Association. Thank you. And we also have membership from Stanley Black & Decker, the Connecticut Department of Environmental Protection, and most importantly, the National Park Services. So we're also honored today to have a member of the National Park Services with us. Andrew Petit-Damage is with us, and if any of you would like to meet him afterwards, it's really the National Park Service is responsible for establishing that designation, funding our committee, and really shepherding us through this whole process. And we kind of really love the relationship because the National Park Service, as you can imagine, is a great sponsor, but they give each of their river committees a lot of latitude in terms of doing what's right and what's of interest for their specific community. So it really, you can kind of see this network of 9 towns, 3 watershed associations, a member, Stanley Black & Decker, because they have the dam and the power generation system down on the Rainbow Reservoir, the Connecticut DEP, and the national parks, right? That integrated community really looking out for the interest of the river. And so what does it take to become a wild and scenic designation? It has to be a river that has some outstanding resource values, right? And when we think about outstanding resource values, they think about the geology, that exists, the water quality, the biodiversity that Dennis is going to be talking about today, the cultural landscape, and the recreation. And if you think about all those components, right, certainly the Farmington River, particularly the lower Farmington River, you see a lot of recreation. My guess is a lot of you are avid kayakers. The cultural landscape is pristine. If anybody kayaks down the river, you probably only see a half dozen homes along the river, or buildings or structures. Not a lot of bridges, not a lot of structures. Part of the charge of our committee, right, is to try and keep it that way, to preserve that scenic beauty. The biodiversity, anybody who's seen any of Rick's photos, you just know it's unbelievable. Every time I go down the river, I think I see something new, whether it's new birds, new wildlife. The water quality, I've lived in Simsbury for over 40 years. The water quality today is probably better than it's ever been, and it continues to improve almost every year. And then the geology. I think particularly when we talk about the upper river and the Salmon Brook, right, it has a very particular geological structure. So with all those qualities, you would have thought it would have been a layup to get the wild and scenic designation for the river, but it was not. Anything with the federal government is never easily achieved, and it really took somebody like Sally to put a committee together and to really stick with this for almost a decade to get this designation. So some of the other, our guiding principles, when we think about the guiding principles of our committee, we've talked about some of this before. It's resource conservation for the protection of the river. It's management of the river through a cooperative approach. So we talked about all the membership groups that are represented in our committee. And we talk about land conservation. So we act in concert with other groups like land trusts, the Watershed associations, Stanley Black & Decker, right, who is a large property owner and has a dam across the river. So it takes that kind of collaboration, a cooperative management approach with a focus on resource protection. Those are kind of all the key themes that are always weaved into kind of any of our conversations at our committees. And then here, just kind of laundry listing quickly. We talk about the other wild and scenic rivers in this area. We talked about there's 10 with such a designation in New England, and then there's a few others here in the east, but you can see the ones in Connecticut, New England, are the 8 Mile River, the Farmington River, the lower Farmington River, and people ask sometimes, why is it treated as 2? It really works out for our benefit that way. If it's treated as 2 separate rivers, you know, we get kind of twice the bang for the buck, and the National Parks treats it as 2 separate entities, so we get additional funding for that. You can see the Pequabic River, the Great Egg River is in New Jersey, but as you go down the list here, you can see that the top 4 are in Connecticut, and where the other rivers with this designation are located, either in New England or in the east. And then, so what have we done so far? As I said, this committee, the river just got its designation in 2019. The committee was finally formed at the end of 2019. Just as we were starting to pick up momentum, COVID hit. And that's why I really kind of am enjoying today. We started our meetings here at the public library. We had about 4 or 5 of them, end of 19, beginning of 2020. And then with COVID, we had to convert to the Zoom meetings. And we've still been doing that, but we haven't let that slow our momentum down. We have funding from the National Park Service, and the things we try and do with that funding are grants that are consistent with our core values. So you can see some of the stuff we've done. We've approved a grant for the town of Avon for the clearing of a trail along the Farmington River through Fisher Meadow. For the town of Bloomfield, we hired a geosystems consultant to create maps for their Farmington Park River, which is really important too. A lot of people show up and they want to explore the river, right? And they're at a new park, but they need a map to help show them around. And a lot of kayakers and canoers, right? We want to promote that recreational aspect of the river, but we want people kind of using designated access ways. So we're doing a lot more publication of trail maps for the rivers and where the points of ingress and egress are to the river. And then for East Granby, we provided some funds for their summer camp so they could do some education with young children about the stream's biodiversity and learn more about the natural ecosystem of the river system up there. And then we've provided funding for water quality monitoring for both the Salmon Brook and the Farmington tributaries. We provided research on the natural invasive plants and species, and that kind of promotes things that Dennis has been doing that you'll hear more about today. So these would be all the kind of things that we're looking to promote. And we have, I think, as some of you, this may be the first time you're hearing about our committee, right? We haven't really gone out and done a lot of self-promotion, but as the committee has come into existence, we're starting to, as you can see, do the stickers, get more visibility. And it's interesting, we haven't completely exhausted our grants from 2001 and 2022 just yet. So we actually have quite a bit of funding available for small grants. Usually they're under $5,000. That would be anything consistent with some of the values that we've talked about. So hopefully that's kind of the end of the introduction here. This is just a picture of some of the activities from the trail clearing, the Salmon Brook Park summer camp up in Granby. But just wanted to give everybody kind of a sense for that this committee was out there, some of the things we do, what we stand for, and what the membership is composed of. So I'll pause there. Appreciate that. I'm going to introduce Dennis now, and myself, Sally, and Andrew will be around afterwards if anybody has any questions about our committee.
Hello, everyone. Just get set up here. Thank you. Okay. For a second, it was only showing up there and not on my computer. Okay, well, thank you all for coming out today. This is a great crowd for a Sunday afternoon. My name is Dennis Quinn. I'm the owner of Quinn Ecological, which is an environmental consulting company that specializes in amphibians and reptiles in Connecticut, in New England, and occasionally overseas in South Africa. I have been working as a herpetologist in the state of Connecticut now for better than 20 years, doing most of my work in Connecticut, but also throughout New England. I've taught zoology, field biology, general biology courses at Naugatuck Valley Community College for 15 years now, and this past year I was appointed a faculty position at Central Connecticut State University, where I teach a course in herpetology. Perhaps one of my biggest achievements, at least I feel, is the recent publication of the new book, Conservation of Amphibians and Reptiles in Connecticut. I was a co-author on this with Dr. Michael Clemens, Hank Gruner, myself, and Eric Davison. These are for sale through the Connecticut Department of Energy and Environmental Protection. And all proceeds of that book go directly to the state of Connecticut's Department of Energy and Environmental Protection's non-harvested game program for support of amphibian and reptile conservation in the state of Connecticut. I'm here today just to talk to you guys a little bit about the work that I did during the 2021 field season for the lower Farmington River. And it's going to be a little bit interesting, I hope, interesting of a talk, because I'm doing it a little bit different than I typically do talks. Usually I do them and we just talk about the species and what occurs where and why. We're not doing it that way this time. I'm taking a different approach. I'm going to explain the actual process that goes through my head in developing a project like this, looking at all the methodologies that will need to go into executing a project like this, figuring out how do we develop species lists for a project like this. So all of these different things are going to tie in together. And a lot of it is going to come directly from this book. It's a book that's primarily based on the conservation of amphibians and reptiles in Connecticut, but we do get into a little bit about the biogeography and other factors which play a critical role in where and when you're going to find species throughout the state. So, Connecticut is home to 46 species of amphibian and reptiles. That's a reasonably large number of amphibians and reptiles considering the size of the state of Connecticut. 19 of these species are common and widespread, found throughout the state. You know, species such as the green frog, bullfrog, like red-backed salamanders, the more common species you would expect to find in any regional park, sometimes backyards, depending on the quality of the habitat. The other 27 of our species, however, are not very common. They're restricted in their distributions in Connecticut. And these restrictions are for quite a few different reasons, which we will get into a little bit. But you can imagine that when developing a study like this, to whittle down a list of 46 species over the course of maybe a week or 2 weeks of surveying time that you have to do a project like this, it's a little bit challenging, and you really have to have a deep understanding of the species that occur in Connecticut, where they occur, and their biology to be able to pull one of these studies off. So the first thing that I do in a study is I define the study area. We need to know where the study is occurring. And that may sound silly because a lot of you guys are probably thinking it's in Simsbury, right? That's not good enough. Simsbury is very geologically diverse, and the distribution of herps across the town of Simsbury is just as diverse. So you really have to understand where in Simsbury you are, and as you guys know, we're within the Farmington River Valley. So that's going to have a whole suite of species associated with it versus a whole different suite of species that are associated with systems like the trap rock ledges that you have in Simsbury, Connecticut. Second, I look at the biogeography, geology, and elevation to see how that might interplay on the species that we're going to want to target for our work. And then I look at what we refer to as the conservation guilds. I look at 3 of the 4 conservation guilds. We're going to talk about all 4 later on in the presentation. But conservation guilds are something that we developed for the release of the book, where we group organisms together based on constraints that drive their conservation. So animals that have similar biological properties which cause similar impacts for their survival, really. So I'll get into more detail on that. But the 3 primary guilds that I use are rare habitats. Are we dealing with any rare habitats in this region? Are we dealing with any early successional habitat-dependent species? Is there early successional habitat on site? And there is. And then habitat mosaics. So that's when you have a bunch of different habitats that are intact and related and unfragmented and lay within the geographic area in a mosaic. So if we look at the study area, I should say that I don't like to find them on GIS this early in the game. I'm showing you exactly how I did this. I do it on Google Earth. It's the easiest. You can zoom in and out. You can put on or off the tree canopy layer. You can gain a lot of information from Google Earth. The other thing that I love about Google Earth is that you can go back in history, way, way back in history to like the 50s. So you can see past, I'm sorry. Whoops. Oh, well, I have to use Google Earth. You guys can use your memories, I guess. But Google Earth shows me the land use history and what happened out there for the past 50, 60, 70 years. And that's going to drive quite a bit what I expect to find out there, because a lot of our species are going to be much more sensitive to development than others. So this project started down in the Curtis Park area, extended north all the way up into Terryville Park. And what? Terryville Park. What did I say? Terryville. Oh, Terryville Park. And then I did go into some of the areas on this side of the river. Some were public access, some maybe weren't. So where are we? My box moved a little bit. We're in Simsbury, Connecticut, and you could see Connecticut's landscape is certainly not uniform, and the distribution of species is greatly influenced by the biogeography, the geology, elevation, and regional climate and land use throughout the state. So for example, you have a suite of species, or at least one species, that is completely restricted to the Long Island coastal lowlands. And does anyone want to wager a guess as to what that species is? We're talking just amphibians and reptiles. This one happens to be a turtle, the diamondback terrapin. So this is our only salt marsh species that we have here in Connecticut. So when I'm putting together my list of species or target species for Simsbury, I'm certainly not going to waste any time looking for a diamondback terrapin because it just cannot be in Simsbury. It only occurs in this coastal region within the tidal areas within the coastal towns of Connecticut. If we're looking at other areas throughout the state that are really key geological formations for the distribution of species, we look at the central Connecticut lowlands. There's a suite of species that tend to occur only at 500 feet elevation or below. There's also a suite of species that occur outside of this area because they prefer to be at elevations of 500 feet or above. So elevation plays a big role in the distribution of species. The other thing is some of the geological features that we have in the state of Connecticut. One major one that's very recognizable is in the yellow here, the trap rock ridge lines. There's a whole suite of species that is restricted or finds their greatest densities in their Connecticut distribution along these trap rock ledges. One, the most famous, is the eastern copperhead. That's the one venomous snake that we have, well, two of the venomous snakes that we have in Connecticut, but the one that is much more common. So let's do some examples just to make sure that we're really getting this, because this is really important, especially when it comes to town planning, when it comes to conservation, not just within your town, but regional planning, regional responsibility for individual towns when it comes to the conservation of species. And this is a great example of regional responsibility. So for example, does the town of Simsbury have any responsibility when it comes to protecting the bog turtle in Connecticut? No. It's regionally the responsibility of these towns, primarily just 3 towns at this point because there's only 3 extant sites left in Connecticut. I know that we have 4 extant sites on this map, but I can tell you that unfortunately over the last 15 years that site has become extirpated. The only reason it is in blue here is because the time frame of data collection for this book fell right in the period where that population went extinct. So it was, when the data started to be collected for this book, still extant, but now it's extinct. Okay, extirpated, not extinct. So, but if you look at this, the bog turtle is distributed along the Marble Valley. And the Marble Valley is very important because the Marble Valley is the bedrock that supports the specialized wetlands that the bog turtle uses. These being fens, calcareous fens. So these specialized wetlands are the only wetlands you're going to find the bog turtle in Connecticut. And you can't find the bog turtle outside of these wetlands, and you can't find these wetlands outside of the Marble Valley, which occurs only in the extreme western portion of the state. So we know we can cross that one off the list. Good thing, because that one's a really extensive survey effort, and it takes a lot of time to look for these little guys. Just a side note, Connecticut's and North America's smallest turtle. Also, depending on what list you're looking at, one of the rarest turtle species in the world. They are state endangered and federally threatened. So it doesn't get any more rare than this. We have 3 populations left in Connecticut, and we know of less than 30 individual turtles that are surviving here in the state as of right now. We're doing all we can to protect them, but it is estimated that Connecticut will be the first state within the northern range of this species that will lose the bog turtle. And the primary result of that is going to be habitat fragmentation, wetland degradation through changes in hydrology, which cause an increase in invasive plants to encroach into their wetlands. And that sensitive wetland can't really sustain any changes to hydrology. So we're not going to talk more about that today because we don't have bog turtles in Simsbury. But if you had more questions about that, I'd be happy to answer them after the talk. Let's look at another example. Should we be looking for slimy salamanders? This is a state threatened species in Connecticut. The simple answer is no, because they're restricted within the Hudson Highlands in the extreme western part of the state again. So this is another example of a species distribution being tied to a geographic feature of Connecticut. Only within 1, 2, 3, 4, 5, 6 towns. We probably can't count Greenwich anymore because that population is probably gone now. It's a highly sensitive woodland species, similar and closely related to the redback salamander, which is interesting because that's the most common woodland salamander species that we have in Connecticut. But this species occurs at the extreme eastern edge of its native range. So when individuals of species occur at the periphery of their range, they have a whole suite of specialized habitats that they need to occupy in order to survive. In this one's case, they occupy core forest, has to be core forest. If it's not, it's not buffered enough, and the habitat will change so much that the salamanders will no longer be able to survive in it. And it has to be with these micro pockets of colluvial deposits, these rock pockets, which tend to be very moist, damp, cool, and have a large amount of duff, which is like leaf litter, and fallen logs. And that's where these guys sustain themselves, in these small little patches. And they only occur within the extreme eastern edge on the Hudson Highlands in Connecticut. So we're not looking for them in Simsbury. That's too bad, because they were a lot of fun. But look at this. Our first few dots now falling in Simsbury, right? The eastern Copperhead distribution. This is tied very closely to the bedrock geology within the trap rock ledge systems of the central Connecticut River Valley. And do you have Copperheads in Simsbury? You absolutely do. Did we survey them for them when we did this study along the river? We absolutely did not. Why? There's no reason to survey for Copperheads because they're going to be at these much higher elevations within these trap rock ridge systems. They will come into the lowlands, but we're so far out of the lowland within the Connecticut River Valley that we're not going to hit the eastern Copperhead within our study area. We might hit it a half a mile away, which we could, but right where we weren't going to hit it, so there was no reason to survey for it. So just that little difference in where you are and that little difference in understanding the habitats and the habits of the species you're looking for really helps you to fine-tune your survey efforts to maximize your efficiency and maximize the amount of money that it costs to do such efforts. Because let's face it, these are, hard to come by projects that don't have a lot of funding, and you don't want to take a lot of funding to do projects like this because it's all conservation work. And at the end of the day, that's what we love to do as conservation biologists. Do we want to go through another one? Maybe I put too many examples on. We'll skip this one. This is the northern spring salamander. This is just one of the salamanders that's distributed in the highlands of Connecticut. Most of the time, they're not found below elevations of 1,000 feet. If we look at the box turtle, that's kind of the opposite of that. Most of the time, they're found at below 500 feet in elevation. Their primary distribution is going to be within the central Connecticut River Valley. And look at where our research site is. Pretty neat, right? This is a forested species that depends on a mosaic of habitats, forested habitats, early successional habitats, and late successional habitats, so shrub-scrub type habitats. This species we did survey for, although I was not anticipating finding it. Does anyone want to wager a guess as to why I anticipated we would not find the box turtle in this area? Think about what I said. It's the only fully terrestrial turtle species in Connecticut. Most of our surveys were taking place where? Within floodplain habitat. So this species usually stays out of floodplain habitats because it's not an area where they tend to survive very well because they're not the greatest of swimmers, and when these areas flood, they get washed downstream. So they don't occupy those areas. And I'll show you the exception in the area that we were at where they may potentially occur. This is the big one. This is what got me excited about the project. We've met all the criteria: a species that is a habitat mosaic one, a species that is tied to riverine habitats, and a species that is found only or primarily within glacial lake, historic glacial lake deposits. And our site fits all these characteristics. It falls within a glacial lake deposit. There's a little river running through it called the Farmington, right? And it has tons of floodplain habitat surviving it and a large amount of intact mosaics of habitats within the system. So this species I was almost going to guarantee was going to be there. And sure enough, first night of surveying, we found this, which is awesome. So this is our first species, listed species that we found on the site. Unfortunately, it was our only one as well. Yes?
So all of this sort of data, what you're showing us here, is it in advance of the work?
Yes. I haven't even called Sally back yet.
Okay. So can I just ask, what is the red, what is the red circle?
This basically just showed red are going to be points that are in Bulletin 112, which was the first herp atlas that was published in 1993 by Dr. Clemens. The blue ones are going to be current site records, so ones that we documented post-1993. You can pretty much, for the most part, assume that all red and blue dots are extant populations. There certainly are some that are not. And most of the ones that are red are where those would likely be, because the blue ones are relatively new records of species. And keep in mind, don't get caught up in dots. Dots are just a data point in time. It tells you nothing about how strong the population is, how sustainable the population is, how fragmented the habitat is around that population. You might have noticed when we looked at the box turtle. How do I go back? Back. Look at all the, people must be like, how can the box turtle be listed as special concern in Connecticut? There's dots everywhere. They're everywhere across the state. They're common. They absolutely are not. Finding one individual is very common. Finding one crossing a road, finding one randomly on a hike somewhere, that happens relatively frequently. However, finding a population that has sustainable numbers with an intact habitat, with a mosaic of different habitats within it, is very, very rare. That doesn't happen very much. There's only a handful of box turtles populations in Connecticut that are actually going to sustain this species for hundreds of years if nothing happens to those habitats currently. Fortunately, a lot of them are on state land, but the dots only paint part of the picture and can be very misleading. So now, conservation guilds. We're still developing our species list. Rare habitats. Are there any rare habitats on the site? Well, we don't have headwater cold streams for the spring salamander. Those are distributed in the northern reaches of Connecticut and the upland hills. Slimy salamanders, we know we're well outside the Hudson Highlands, so we don't have any of that rare habitat. We don't have fens because we know now that they're associated with calcareous bedrock. And we don't have any of the diamondback terrapins because we know that they're only associated with the coastal regions of Connecticut. So if we look to see what we've got, we do have one. We don't have the Connecticut River, which is the primary river of the mud puppy, but we do have the Farmington River, which is a tributary stream of the Farmington. And we do know that mud puppies congregate at the mouth of the Farmington and Connecticut River. And I am happy to announce that we will be doing additional surveys for the mud puppy in the Farmington River this coming field season. We've currently been able to document them as the river flows 2.5 miles up into the lower Farmington River from the mouth of the Connecticut River. And there may or may not be a student in the audience today who's working on that project that doesn't want me to mention her name. So, I will say this, we did not survey for mud puppies during our study, because believe it or not, last year we didn't think that we would have got them that far up the Connecticut River. In all honesty, this whole mud puppy project and reevaluating the distribution of the mud puppy in Connecticut started out of complete boredom during this winter. And my buddy and I just started going out into the field, and we were like, well, let's just go look for mud puppies. It's the only species that is active right now, so let's go find them. And we started searching in the Farmington, and we found them like right away. So we kept moving further and further up. Then we went into some of the other tributary streams, and we started getting in those tributary streams. So now we're going to be reevaluating all the tributary streams and some of the other major river systems in Connecticut, like the Thames River, Naugatuck River. We're going to be looking at the Chippewa River and some of those other streams that we have in Connecticut where we don't think that they occur, but they've never really been surveyed for appropriately in the past. So we're going to be rewriting the mud puppy account at the end of this year. Just kidding. Pretty exciting. On the sad side of things is that the mud puppy might not be a native species. It might be introduced. Next year, we're going to be doing some genetic analyses to see if we can determine whether or not mud puppies in Connecticut are native or introduced, which it's suspected they are. They currently are listed as special concern in Connecticut, but they really acquired that listing to drive research to determine if they're native or non-native. So once we determine and answer that question, they'll be taken off that list for sure, unless they're native. So, species that depend on early successional habitats. There's a whole suite of species that depend on early successional habitats. Northern leopard frog, early successional wetlands. Fowler's toad, early successional scarified sandy habitats. We don't have any of those on site, so we're not going to find any Fowler's toad. Five-line skinks, which are early successional habitats, but rocky outcrops. We're not going to find any of those as well, although they do occur in Simsbury. They're just not going to occur where we are surveying in Simsbury. And then the smooth green snake, I was kind of on the fence about. Maybe they'll be there, maybe they won't be there. Some of the habitat's there, some of it's not there. We didn't survey them for them though. Reason being is that green snakes are the most elusive of all the snake species in Connecticut. I'm lucky if I find one a season outside of like my sites where I know they occur in high densities during my survey efforts. Probably not even one every season, probably one every 2 to 3 seasons. In reality is how many I find. So they're very hard to survey for and fun, but you waste a lot of time looking for them. And at the end of the day, the protection and management strategies for a lot of these other more easily found species will ultimately cover the green snake in the end, so you don't have to waste your time looking for them. Don't tell companies like Eversource that, because I still like to look for them on Eversource easements. For whatever reason, I find them a lot on Eversource. This is being live streamed, someone said. I really don't care what they think. So I put kind of an X over that one. Like maybe it's there, but we didn't look for it. And then we have our habitat mosaic dependent species. Of course, these are our mill salamanders, which we surveyed for. I put Jefferson up there, even though I knew we weren't going to get Jefferson. The only reason I put Jefferson is because in order to tell if you have a blue spot or a Jefferson population, you really have to do a genetic analysis. In the field, though, I would know pretty much that we were looking at blue-spotted salamanders primarily because we were in a riparian system. That's where blue-spotted salamanders breed. Jeffersons breed more in the ridge lines, in the more high elevation areas. But they do have genetic influence on each other, so you end up getting hybrids. There are no pure diploid blue-spotted salamander populations in Simsbury or west of the Connecticut River, and we have no diploid Jefferson salamanders anywhere in Connecticut. They're all interbreeds between the blue-spotted and the Jefferson salamander. You can get diploid or parental populations of blue-spotted salamander in extreme southeastern Connecticut within the Quinnebog Valley. Most turtles, wood frogs, and snakes, all habitat mosaic-dependent species. So we looked for all of this stuff. What is a habitat mosaic-dependent species? This is a species that uses a variety of different habitats throughout the season. It also makes them very vulnerable to a lot of external stressors, and they really require intact mosaics of habitat, because once you fragment them and put roadways to their habitats, they start to die off very quickly because of direct impact impact to their habitats and road mortality and everything that goes along with that. But if we take this example, which comes right out of the book, the spotted turtle emerges in the early spring. Some of my stuff is cut off down here. There are letters down here, but, or something. They emerge from their forest, their shrubby wetlands where they hibernate.
Because I don't know if there's any batteries for that other one.
And then they move into vernal pools in the early spring. The reason they do this is it's a great foraging opportunity. They're going to go in there and
Go ahead, yes. Yes, 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 yes.
And juvenile wood frogs and juvenile salamanders. From there, you're going to get a couple of different movements. Males may stay here, but females are going to move into these early successional habits. Did I not turn it on this whole
No,
time?
I'm the one who didn't have it on, but you're.
All right. Hello. So the females will move into the early successional habitats to nest. Some males may stay within vernal pools, especially if they haven't dried yet. And then as you get into the late summer months when it's really hot and really dry, they move into these forested areas on the edge of their overwintering wetlands, and that's where they stay estivate. They actually, a lot of times, will dig themselves in underground to wait out the really hot and dry months of the year, and then they'll reemerge, go back into their shrubland swamps, walk some around for a little bit, hunt a little bit, until they drop back into hibernation. So they have a pretty interesting life history, pretty complex life history, and because of that, they're using a wide area on the landscape, which makes conservation of this species and a lot of these habitat mosaic species extremely challenging. So, after all that, I came up with 8 species. Well, I should say 8 species of conservation concern. I'm always looking for the common ones. Jefferson salamander complex, blue-spotted salamander, northern leopard frog, spotted turtle, wood turtle, eastern box, eastern hognose snakes, and the eastern ribbon snake. So now that we have this species list, how are we going to maximize our time in the field to be able to adequately survey for these species to determine whether or not they're actually on the site? Well, I have 6 primary methods that I use. Number 1 is diurnal visual encounter surveys. These are where we go out during the day and just visually look for animals that are out in the field. I find that diurnal visual surveys work better in the summer months, spring months and summer months for reptiles, and nocturnal visual encounter surveys, this is when we go out at night and do the same thing but with headlamps, work better for the amphibians during the early spring season. So I'm spending most of my early spring nights in the field and then switching to daytime field work later on in the season when it starts to warm up a little bit. We do call surveys primarily at night, but also during the day, because most frogs are calling at night, not during the day. Although you do hear them during the day, they're just calling a lot louder at night. And then we use non-baited minnow traps. Non-baited minnow traps are used to catch the embystomid salamanders, like spotted salamanders and blue-spotted salamanders. We come back during the day to do egg mass counts for salamanders and wood frogs, leopard frogs, so we could see who's been breeding where in what relative abundance by the number of egg masses they lay within the wetlands. And then we do, later in the season, cover object surveys to look to see where snakes might be hiding out, things like that. And that's when we combine our cover object surveys with our diurnal surveys during the day. Any questions on any of that? I think I have to hold it for the streaming. I'm sorry. I know it's like awkward. Does anyone know what these are? Egg masses from the vernal pool of a spotted salamander. So these are spotted salamander egg masses. This is my team. This is what we look like outside in February and March when we're freezing. This is my lead herpetologist, Jeff Cavallaro, who couldn't be here today because he contracted COVID. Don't worry, I haven't been with him in a long time, so I don't have it. And then we have a couple of students of mine, Tiana and Kirk, who come out. when we're out in the field doing these nocturnal surveys, we're using high 1,000 lumen high output LED headlamps. These are very, very bright. They would light this room up. From here, you would light the entire room up. They're extremely bright. And the reason we do this is because it lights up all the eyes of the frogs. It's the preferred method to see frogs. You can detect frog activity from about 100 meters away using this methodology. So you can see a bunch of different frogs across the landscape. We also use these in the wetlands. Unfortunately, you can't shine salamander eyes. They don't reflect back. So we just shine our lights into the pools and you can see the salamanders swimming around at night, which is the advantage to being out there at night versus during the day, because during the day, there's no activity in the pool. You might have wood frogs calling, but you're not going to see active salamanders moving around. You will see egg masses, but it's always more fun to see the actual animals. This is what a night out would sound like for us. Here in the background, a bunch of wood frogs. In the foreground, spring peepers calling. This is filmed right down the road. This is at the site. So it's pretty loud. I mean, it was pretty loud in here, but when you're actually in the field, it's deafening loud. You can't even talk to each other. We're screaming across the wetland at each other. I don't know what just happened there. And I have one more. And I want you to listen very closely. This is the leopard frogs calling, but they're hard to hear. Hear that in the background? That I can't do it. The peeping is spring peepers. The leopard frog is in the background. It's like, oh, 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 oh. That's not what it sounds like, but I'll play it one more time so you can hear it. Hear it? And this is right down the road. Minnow trapping. So this is minnow trapping. We use this for primarily our mole salamanders, Jefferson's blue spots, and spotted salamanders. This is Tiana putting together a minnow trap. This is what they look like. For the embystomid salamanders, we use non-baited ones because we don't want to bring predators into the pool inadvertently to try to drive the salamanders into them. They get stuck in them and trapped in them on their own, so we don't need to bait them. However, when we're doing our surveys for mud puppies, we do bait them, and we bait them with beef or cow liver. Wait, that's the same thing. Two different kinds of livers. I forget what the other one is now off the top of my head. What is the other? Go? I don't know. Anyway, this is beef liver. So we bait them with liver, and then we let them soak overnight, and we come the following morning to check to see if mud puppies had gone into the traps. And most of the time, they do. So that's a great way to find mud puppies. And this is the Farmington River. If you've noticed the beginning of the survey efforts in the Farmington River, you may recognize this bridge abutment. That's the one that the train goes over. I forget the name of the road. Sorry. So all this time and effort, what did we do out there? So we started our field surveys on March 12th. We're just a visual assessment of the site to help guide our nocturnal survey activities to see where we needed to go. If you go out to a site for the first time at night, you're going to get lost. You're not going to be productive. You really need to do a day ahead of time to make sure you know where you're going at night. Then we started our actual surveys on March 24th. And we put in, throughout the entire time, a total of 39 survey hours and 112 man-hours of surveying for amphibians within this stretch of river. We also did a total of 71 survey hours for our reptiles. Wait, that's not right. 32 surveys for our reptiles, totaling 81 person hours, and a total of 71 overall survey hours with 193 overall man hours. So we spent 193 hours surveying for amphibians and reptiles within about a 1.5 mile, 2 mile stretch of river. And we did this seasonally, as you could see, starting in the beginning of the amphibian season and continuing in the beginning of the reptile season, ceasing activity in June, around June 15th, right or prior to the nesting activity of a lot of our turtle species. But this 2021 season was a little bit strange, and it wasn't very conducive to surveying for turtles because the early spring and early summer were so cold, and then it warmed up really quick. And by the time it warmed up, all the plants had already grown in. Like, the plants grew before the turtles came out, which made it very difficult to document them because they went right into really thick cover right off the bat last year. But we did put together a list of species. We got a couple of our salamanders, quite a few frog species, a couple of turtles. Unfortunately, no spotted turtles, but I know that they're there. We just didn't get them. They have to be there, especially on the west side of the river. And then we got a couple of snake species. We got the relative of the listed ribbon snake. We got the eastern garter snake, but I do expect that the ribbon snake does occur on site, even though it went undetected. But the one that we're going to talk about now, as far as conservation goes, is the northern leopard frog. I'm not sure how much time I have left. Does anyone know? 15. All right. So, how are we going to conserve the diversity of amphibians and reptiles found within the study area? Outlined in blue here are the 3 areas where we have documented leopard frogs breeding. So these are going to be our 3 key areas as far as protection goes. And you can notice, although it's relatively unfragmented as a whole for Connecticut, this site is still highly fragmented. So it's facing a lot of pressures already. So it is certainly time to act, because if we don't act now, there's going to be even more pressure and more declines on this species in this part of the river moving forward. So, understanding species and conservation challenges. This is where we talk about the conservation guild approach, which I've talked quite a bit about already. But the foundational concepts are the biological and ecological constraints of species, the anthropomorphic stressors acting on species and populations, and then the synergistic effect and interplay between A and B. Okay? So that's very important. Again, we break it down by guilds and the conservation challenges associated with each of those guilds, and then the critical roles that stakeholders play, especially NGOs and municipal governments. A lot of people think that conservation really takes place at the state government level. It really doesn't. It doesn't really happen at the state level. It doesn't really happen at the federal level. It does in certain circumstances, but not all. Where most of the conservation is occurring in Connecticut is at the town level within one of each of our 169 towns through planning and zoning commissions, inland wetlands commissions, and places like that. That's where decisions are being made. If you want to have your biggest impact in conservation in the environmental science field in Connecticut, join your local inland wetlands commission or planning and zoning commission, because that's where your voice will be heard the most. So these are our guilds again. The only one we did not talk about are long-lived species with delayed sexual maturity that have a low fecundity. And these are primarily our reptiles like our turtles and our timber rattlesnake. So I talk about this because I do suspect there are probably some wood turtles within this river, although wood turtles don't really like the Farmington River too much. I don't know why. It's a beautiful place to live. I think it's just a little too big for them and a little bit too deep. They tend to be more in their tributary streams, and they tend to be a little bit further up, but it's okay. Maybe they'll work their way down. But if we look at the conservation guilds, you could see how these individual species fall and how they break out into guilds. And what I haven't mentioned yet is that, this is very hard to see the colors on this, by the way. I could barely see it. Could you see the differentiation of the green, blue, and purple on that slide? Okay. What I did not mention is that a single species can be in multiple guilds, which means it has multiple stressors affecting it. So if we look at something like the eastern box turtle, it's an early successional habitat-dependent species. It's a habitat mosaic dependent species, and it also is long-lived with delayed sexual maturity and low fecundity. So all of these factors playing together and all the challenges within each of these guilds is going to play on the conservation and survivorship of the eastern box turtle. I'm not going to get into extreme detail on that. It's in extreme detail in the book. But we know we have our species that depend on rare habitat, the mud puppy, so we should be looking at conservation solutions for that species within the Farmington River. We know that we have a few species that are dependent on early successional habitat, so we should be looking at species or conservation methods that would improve the habitat for these, specifically the leopard frog. And we should be looking, because we know that we have a couple of habitat mosaic dependent species, specifically the wood frog. We saw those out there, so we should be looking at conservation strategies to promote the success of that species. We have no long-lived reptiles really out there. We do have painted turtles, so that's a good thing. And this is really going to interplay with some of the other ones, so we're not going to talk too much about this one today. But what's critical to understand is that species are not equally vulnerable to stressors. They're just not. So we have here a northern leopard frog, we have here a green frog. They're both frogs, they're both aquatic species, but they are not equals when it comes to how they respond to stressors. Stressors are variable. It can be habitat loss and fragmentation. Green frogs can often thrive in those sorts of conditions. But leopard frogs cannot thrive in those sorts of conditions. What's one of the reasons they can't thrive? They're a habitat mosaic dependent species. They need to move between wetland habitats and upland early successional habitats. And once you fragment those habitats from one another, you're going to be creating areas where they're going to be getting killed off pretty consistently. Versus green frogs, which tend to be pretty sedentary in the ponds that they decide to reside in. Once you do have habitat fragmentation, you have a whole suite of other factors that start to happen. You alter your hydrology of your wetlands. You initiate the invasion of, or the encroachment of invasive species. Look at things like chemical contaminants. They're on the increase, they're on the rise. This is all salt. This photograph was taken 3 miles from here. This salt was washed right into the Connecticut River, right at the confluence of the Farmington River. Is there really a need for this salt this time of year on the sidewalk that no one's using? You guys be the judge, not me. We know that these salts have a really devastating effect, especially on leopard frogs. They're increasing the salinity of wetlands. They're degrading the habitats. Leopard frogs are highly specialized. I know salts don't govern pH, but complexes of different salt ions can impact pH. And what we're starting to see is increases of pH within our wetlands. And leopard frogs can only successfully breed in wetlands that have pHs of between 5 and 6 and 7. Once you start to get into the 3 and 4 level, it starts to kill off the egg masses and the juvenile tadpole stage of the leopard frogs. It also causes less fertilization to occur because the sperm of the male become less mobile in acidic waters. So it's very important that we're looking at these things and how they're impacting the water chemistries for our resources that are using these wetlands. We are losing, and this is not unique to Connecticut, we are losing a lot of leopard frog populations, especially in the southern or lower part of the Connecticut River Valley. So when we're studying new development, I'm going really quick now, we have to plan at multiple scales. We talk about the macro scale, the meso scale, and the micro scale. The macro scale is all the green. At a minimum, you really want to see 1,000 acres of habitat that's intact to protect these species, ideally 2,500 plus acres of habitat. The meso scale is the development parcel, so that's all this stuff that's in black. You want to do as much as you can in here to help promote conservation, but your efforts are really not going to go very far because there's already going to be impacts bleeding out from the actual development footprint, and that's the micro scale. That's the individual lot/site plan. So you have this roadway coming in with this housing development, which is going to be impacting not just the habitat directly, but indirectly way out into all of this habitat out here. So we need to understand that development isn't a localized impact, it's a regional impact, and it has implications much further and farther beyond the edge of development. So this is our study site. The number one thing that we need to do, and this is a critical stage in conservation, is the fact that this unfragmented parcel is approximately 700 acres, which means what? We're already below the threshold of 1,000. That's a problem. Can we catch it in time? Well, if we're going to catch it in time, and if any of this work is going to make, for a better population of leopard frogs in the long term, we need land acquisition. If we don't have land acquisition, over time we're going to lose this leopard frog population. And the more and more development that goes into these areas, the quicker that loss will occur. Fortunately, this parcel is available for purchase if anyone is interested. It actually houses one of the biggest breeding area that we saw on the site. And I joke, but the Connecticut Department of Energy and Environmental Protection saw about this study and is actually in the process of pursuing this land for acquisition to put it into management. So land acquisition is number one. We need to do that. Absolutely need to do that. We need to do habitat management for invasive species. So where we have habitat in here, we need to make it better. We need to get rid of our Phragmites. That's ruining our wetlands. We need to get rid of our things like multiflora rose. Multiflora rose is really bad for nesting turtles because it causes the successional process to happen very quick, shading out nesting areas that turtles rely on for reproduction. We need to get rid of things like Japanese barberry. Japanese barberry is a species, once it starts to form monocultures in forested habitats, it's known to cause declines in amphibian abundance and diversity. I can go on all day about this, but I'm running out of time. Next, we need to, land acquisition is number one, so I already said that. We need to talk a little bit more about reductions in chemical contaminants. I'm not sure what the process is here, but I see a lot of playing fields. I see a major road. I'm assuming this road is salted and sanded. I actually know it's sanded, so don't lie to me. I saw the sand. But I'm assuming that it's also salted in the winter months, which is, do we need it to be salted when it's falling right next to the Farmington River, which is draining right out to the Connecticut River? We have enough issues right now with the fragmentation of the landscape instead of putting in additional chemical contaminants that increase the stressors on a lot of these species. And I don't mean to be mean to your parks, and I think you should keep them, but I think we could do better. If we look at a situation like Curtis Park, we have a lot of habitat along this area here, but it's bordered by forest, and then a clean cut right into your playing field, your soccer field. This is fine. We should have soccer fields for the kids to get them outside, but little things like 10 to 15 foot early successional buffers can make a big difference for the conservation of these species. It allows leopard frogs places to forage, places to seek shelter, and they'll ultimately use the actual playing field itself much less. Because remember, this is an early successional dependent species. It needs early successional habitat, and it will find it. And on one of our first leopard frogs that we found was out on the ball field. We've caught probably 20 plus leopard frogs every night on this ball field. I'm not saying that's a huge issue because we're not out there cutting the lawn at midnight on a rainy night. Probably not doing that anyway. If we are, that is a problem. But we want to be sure that we're creating more suitable habitat, reducing their exposure to predators and things like that, affording them cover in areas where they can actually forage more safely. And when we're maintaining these, we want to reduce the use of pesticides, herbicides, which we're probably doing already because they're ball fields for kids, I would hope. But maybe we're not. And when we're mowing, even strategies for mowing can help, just a little bit. Don't mow around the edges to start if you're going to do that. Start from the middle and mow in concentric circles out to the edge. That will give the animals using the field plenty of time to evacuate the field and move into the surrounding habitats. This here really is what bothers me the most. This can be converted into early successional habitat, and ultimately should be. Nowhere along the Farmington River within a park system, in my opinion, should there be open lawn up to the edge of the Farmington River. It should not be happening in this day and age. So, with that being said, before I get kicked out, just thank you and happy National Frog Day.
We've got a few minutes. First of all, let me say, Dennis, terrific presentation. I don't know about you guys, but you know, when I see a thing about herpetology, and is that really going to get me enthused? I kind of scratched my head and said, are people really going to turn out on Sunday afternoon and hear about reptiles and amphibians? Well, when you've got passion and you've got knowledge like Dennis has, it's enough to bring me out a lot of Sunday afternoons. So I want to
thank you. I appreciate that.
And please, any questions?
Yes, questions?
What I want to tell you that I'm very pleased with is that in that area where you pointed along the lawn in Curtis Park, one of the things we requested as the Lower Farmington River and Salmon Brook Wild Scenic Committee through our National Park Service was some tree planting along there in relation to a trail that the Army Corps would permit. And so the National Park Service had the opportunity to comment. And that was one of the things we included.
Excellent. Excellent. That's perfect. The frogs will be very happy to see trees and shrubs and longer grass in that area for sure. Any other questions? Yes.
What are mud puppies?
Mud puppies are Connecticut's largest salamander. They're fully aquatic. They retain their gills into adulthood. That's referred to as paedomorphism, where you retain juvenile characteristics into adulthood. They reach about 17 inches as adults, and they're just in the Connecticut River and really the Housatonic River, the two main stem rivers that they're in. So their distribution is pretty restricted in Connecticut. It's just a really large salamander. Okay, question.
When they're going to the vernal pools and they have to cross roads, are there volunteer brigades that go out there?
I can answer this one for a while. There's been an increase in what are called, well, it's called the big night, when all the salamanders and frogs come out to migrate. And there's been this big trend to set up these brigades of people to help shuffle frogs and salamanders across roads. That is a really great and a really nice gesture. But I think we need to come clean and be abundantly clear that that is not a conservation strategy. That is something you do to feel good about yourself at the end of the day. There is data that shows, even in these pools where these brigades are crossing salamanders over, we're still seeing a net decline in populations. Reason being, you're getting a salamander one time going into a pool, the brigades are gone by the time the salamander's migrating back out across the road, and we're missing the entire migration of the juvenile salamanders from the vernal pool out into the forest later in the fall. So you're catching a one moment in time, and you're feeling really good about it, and we're spreading this as, oh, we're doing so much for conservation, but I'm starting to feel that some of this promotion of these events is hurting conservation in the end, because it's giving people the misconception that they're doing everything they need to be done to appropriately protect these species, and they're not. Don't stop doing it, though.
Do you think those roads should be closed during those times?
I think there's certain roads that should be closed during those times, but once the road is there, the damage is done. What we really need to do is get back to that 169 towns idea. Get into your local municipal councils and vote against the development of these new roads when they're not cited with sound science. If people are not going out there and conducting the appropriate surveys, meaning looking to see where the vernal pools are, looking to see where the forests are, where these salamanders are using, we should not be building roads anymore in between vernal pools, wetlands, and migratory corridors for amphibian wildlife. That's what we need to be doing. Until we stop doing that, we're only going to be causing net loss and declines of amphibian populations statewide. Yeah.
So I noticed you mentioned a lot about turtles. Does the musk turtle occur in this Farmington Valley area?
Yes, the musk turtle is actually in the Farmington River. Oddly enough, the musk turtle is like one of the only turtles that isn't considered a habitat mosaic species. Usually all turtles are, because they're going from aquatic habitats to upland habitats to lay their eggs. The musk turtle nests basically on the banks of the river. So it never really leaves the main corridor of the river itself. It's never going outside of the upland review area of the river. So we don't consider it, although it is nesting on land, we don't consider it a habitat mosaic species. But yes, it does occur in the Connecticut River. It's a riparian species that occurs pretty readily throughout the state of Connecticut. Yep.
I have a question about turtle nests. When I find turtle nests and they're open, the eggs are open and laying around, is there a way to tell if those have been preyed on or if they've hatched?
Yeah, if you're seeing the eggshells, they were preyed upon. You will never see the eggshells in a nest that hatched naturally. What you'll see is a little hole in the ground where all the babies have crawled out of. If it was dug up and the eggs were eaten and splattered all over and tossed all over around the nesting cavity, you know that those eggs were eaten. It's I've in my entire career have never come across an actual hatched turtle nest. I only see them when I cage turtle nests for various projects I'm working on. Yep.
Yes, Dennis, how do you get the information that you've imparted today to us, concerned citizens, to the people in these various towns, like in Simsbury, that actually do make the decisions on development? I mean, do you talk to the inland wetlands commissions of various towns, try to educate them because all the towns are being pushed for development? Yeah.
Yeah, you're absolutely right. And I will talk with anyone who wants to listen. I'm not often invited, but I will talk. You'd be surprised the politics that occur in some of these commissions. I'm not going to talk about that here today. This is live streaming. But it's all outlined in the book. There's a whole entire chapter on municipalities and the role that they play in conservation. We're hoping that this book gets out to them, those organizations. We do talk to them. There are certain towns that are more conservation minded than others. It would be great if people in this room can help spread the message. I think one thing, which not to offend anybody, but I'm pretty good at that, and I'm going to say this anyway, if we look at the demographic in this room today, we should all be a little bit upset by that. This is mostly older individuals. Where's our younger generation? The ones who are going to be right here, one person, 2, 3, 4. Very few people here that are going to be taking over for everyone in this room in the following 10, 20, 30 years. Where are those people today? Those are the ones we need to be reaching out to, or we're going to continue to go backwards in the field of conservation, especially here in Connecticut. But thanks for coming.
One more question that she really wanted to ask is about a citizen survey, like we do an Eagle survey in January. Are there any such for this?
There are certainly citizen opportunities, citizen science opportunities with the state and various organizations. There's one that you can look up now. It's called Frog Watch. That's happening now. I think it's done through Norwalk Aquarium and Mystic Aquarium, where they have volunteers. They train them how to identify frogs and frog calls, and then you go out at night at a designated area that they tell you to survey for and report back the data. There's usually opportunities if you contact the Connecticut Deep for various projects that are going on. You can reach out to me, and I can get you in contact with people, or you're welcome to come help me and volunteer on some of my efforts that I'm doing in the field. So, one more question, or is that it? There's one more. One more. And then we're...
2 questions. One is, do you do presentations in schools? And the second one is, I didn't hear anything, but curious sort of about the daunting issue of climate change and how that will affect your work over the next few decades.
I'll be out of business. I do occasionally do talks for school groups. Yes, I do maybe 2 or 3 a year typically. But climate change, yes, that's a whole issue into itself. I certainly can't go into much detail. But, you know, ultimately, I think the best thing that I can say about that right now is ultimately we don't know. We don't have the data. We're starting to see some things start to come out impacting wetlands. impacting breeding amphibians. We're seeing some species respond well to it, some not responding so well to it. You have to figure that a species like the spring salamander, which is in headwater streams, relies on very cold water shaded conditions, the loss of the hemlock tree, increasing water temperatures. That species is probably going to retract its range out of Connecticut. It's probably not going to grow its range, versus other species will probably benefit from climate change. Overall, I would say, you know, especially with turtles and temperature-dependent sex determination, how is that going to influence sex ratios down the line? There's been a study that showed that older female snapping turtles have, over the last a few decades, adjusted when they lay their eggs. So they're laying their eggs later in the season. Older ones, the younger ones, the new generation, is right on schedule from where they were as if climate change never happened. What's the consequence of that? What's the consequence of a species like a painted turtle where the juvenile overwinter in the eggs? What's the fitness consequence if they do that for too long and don't have the resources when they hatch in the spring to be able to survive? There's a lot of unknown questions. It's really a hot spot right now, and we need a lot of research. The problem is we're working with long-lived species, and you need decades of data to even answer the simplest of questions when it comes to climate change. But it's something we all should be concerned about and doing something about, for sure.
I'm going to ask, Dennis is going to be here for a minute, so if you guys could hold your questions, we're going to sort of get this moving quickly. We couldn't be in this room, and in fact, we scheduled it. We wanted to do this program in March because the Farmington River quilt is in here. And we couldn't let that go by without some explanation of it. So I'd like to introduce David Seinish. Just a couple of quick data points. David is an active member of the Farmington River Watershed Association, and has been since 1981. He was very active in helping achieve the Wild and Scenic designation for the upper Farmington River, and he's on the Farmington River Coordinating Committee, in addition to being an avid kayaker and just a real tremendous voice for the river. So David's going to talk to us a bit about the quilt that you can take a look at over coffee and a cookie afterwards. Thanks, David.
Well, thank you for that introduction. I have to tell you that my mind has really been opened with that previous presentation. That was great. 429 words. I have been a director or officer of the FRWA since 1981, and since then I have become very involved with the Wild and Scenic designation of the upper Farmington River, serving primarily on the Education and Outreach Subcommittee. In 1981, the Metropolitan District Commission had a referendum to move Farmington River water from the west to the East Branch Reservoir system. This action would possibly dewater the river downstream of Barkhamsted. So you think about that long length. FRWA led the opposition to the referendum, which was voted upon by MDC service towns and not by the towns along the river. The referendum was defeated, and FRWA wondered what could be done. The Wild and Scenic Rivers Act had been passed in 1968, but had been used only in the far west. Could it work here? Many meetings and much study later, the Upper Farmington was designated in 1994 as the first federal partnership Wild and Scenic River. The partnership is between the 5 riverfronting towns, other organizations with a strong connection to the river, and the Federal National Park Service. You can read the complete story on the FRWA website. Go to River Info, Farmington River Wild and Scenic. And the author of that is speaking to you right now. So I very much recommend it. The lower Farmington and Salmon Brook Wild and Scenic River was designated in 2019. Wild and scenic rivers are judged by the outstandingly remarkable values of recreation, fish, wildlife, scenic and historic resources. Coordinating committees provide oversight to ensure that river activities are in keeping with the federally approved management plan. These coordinating committees have members from all riverfronting towns and other interested entities. These subcommittees are usually resource stewardship and education and outreach. I serve as chair of the Education and Outreach Committee. My committee works to get the word out about the beautiful, wild and scenic Upper Farmington River. Our thought is, if they know it and love it, they will protect it. Now, remember that one of the ORVs, the outstandingly remarkable values, is scenic. There is an extremely talented quilter who lives in Winchester, Connecticut, and loves the Farmington River. In one of her travels out west, she saw a quilt of a section of the Sacramento River. She was inspired. She came to the E&O committee and volunteered her service and those of her quilting friends to showcase the Upper Farmington River. Mary Pat Leisure, what a gift you have given to us all. The quilt is 50 feet long, has 25 panels, and is the work of 24 different quilting artists. The first panel, way over in the corner, depicts a scene in Colebrook. And the following panels trace the river flows down to Canton. Each panel has attached comments from the artist, and there are some comments about that specific location. This gorgeous quilt has been hung in many different places, with perhaps the best in the walkway between the Hartford Capitol Building and the Legislative Office Building, on the long wall by the escalator. And if you've been there, you realize that that escalator climbs up maybe 20 feet, which is a perfect spot to showcase the river flowing downhill. Now, today does not have to be your only view of this quilt. If you Google the Farmington River quilt, there are pictures of every panel who have been photographed by Tom Cameron. And thus endeth the reading.
Terrific. Thank you, David. Really appreciate it. I think we think about the work that went into the quilt that's around us, and we think about the work that is yet to be done to make sure that we are preserving the river that we have that runs through our town. It really is it's a treasure. I want to thank the Lower Farmington, I'm not going to say the whole thing, the Lower Farmington Wild and Scenic Committee for sponsoring Dennis's talk. Dennis, thank you so much. Really tremendous and very informational. And you know, the number of questions that come from a presentation are an indication of the interest. So, you know, very, very well done. And thank you very much. And David, thank you for giving us insight into the Farmington River quilt. It's wonderful. On behalf of the Simsbury River people, thank you all for coming. We hope you'll stick around. There are a lot of folks in here who would be happy to talk to you, provide answers to questions, or get them to you if they don't know. So thank you all. Have a great Sunday afternoon. Happy spring.
Funding for Simsbury Community Television is provided in part by contributions from viewers like you. Thank you.